Is there a specific species of algae that should be used to create the biofuel based on your research?
This is a broad classification. Under freshwater, you have different strains, different species, and different families as a plant kingdom. So we have for algae as well. So the similar thing applies to marine microalgae as well. So if you're concentrating on biofuel, the best candidate so far identified for biofuel is Cynodesmus obliquus. That is the best candidate which has been identified for biofuel production.
Okay.
Yeah. So if you're looking at some other algae, there are a lot of algae which have been researched on for producing biofuel, but the best candidate so far identified is Cynodesmus obliquus.
Okay.
And does this species of algae grow in North America? Is it everywhere?
I'm not sure of whether it grows in North America, but what I did was I obtained it from culture collection of Algae and Protozoa from the UK.
Okay.
So they have a variety of collections of algae, like whatever algae they discover, they have to deposit in. They deposit in like all these culture collection stuff. Like they have a collection of algae. So there's one more in the US. Like Texas University has one more collection where you can get some algae, as well as in Canada there are some universities which do have some collection of algae, but I'm not sure like whether they have Cynodesmus. But I got it from UK, Center Culture Collection of Algae and Protozoa.
And what are the specific characteristics that make this species of algae the ideal candidate for biofuel production?
Specific characteristics, because when you grow it under nitrogen deprivation, there's an increase in lipid production. That's what I've seen. I guess like that's what I've read through, but that's the major reason. So if you enhance your lipid production, obviously your biofuel is going to get enhanced. So that's the main goal of.
Basically what he told us is that the more lipid an algae has, the better of a fuel it will be. And to increase the amount of lipid an algae has, he made them grow under nitrogen deprivation.
What is the simplest and most effective method to extract the biofuel?
Like transesterification is the most important commonly used method for extracting biodiesel. And for characterization, you can use GCMS, like a gas chromatography, where you can just get the amount, what are the total saturated and unsaturated fatty acids based on. So that's the major thing. Okay. So transesterification is the major.
And what kind of equipment would you need to do this?
Just normal chemicals in the lab for transesterification. You don't require any instrument like hi-fi instrument. But when you're analyzing for the saturated and unsaturated fatty acids, you require analytical instrument in order to analyze it. Like, okay, this is the percentage of saturated fatty acids, because there are a lot of fatty acids which are responsible for biofuel production. So,
what are the optimal conditions for this specific species of algae to grow?
So far, as I've researched, the optimal temperature for growing it is between 14 to 30 degrees Celsius. Okay. So you can grow anywhere between 14 to 30, but the optimum would be around 25 to 28. So that's the optimum temperature. But you can grow between 14 to 30, and also it can survive extreme of temperatures. It can go up to 38 to 40. So it can go up to 40 as well. But your biomass productivity would be low. So that is as far as temperature is concerned. As far as light intensity is concerned, it can go you can use anywhere between 40 to 100 micromoles. So that will be like the normal thing. I'm using 40 micromoles because it's a lower light intensity. 40 micromole itself is a lower light intensity. I'm using 40 micromole because I'm using it for 24 hours in order to save up energy. So I'm running it for 24 hours, so I'm keeping the intensity low. Whereas if you're using it like a day cycle, like a 16-8 or a 8-16 or like whatever you want, 12-12, in that case you can go for higher light intensities, and also you can reduce the number of photo period as well, because you're reducing the photo period, right? So if you're going for a 12-hour light and a 12-hour darkness, you're obviously reducing energy in that case. So it depends on what you want.
Okay. In our research, we've encountered the hexane solvent method. Is it productive?
As far as my knowledge goes by, hexane is also like one of the best solvents to be used for biofuel production. But whereas you can also try other solvents, like if you're looking at like optimal biofuel production, there are other solvents as well. So
like what? Do you have any?
No, I have no idea. But I've gone through it, but I don't remember anything on top of my head. I'm sorry.
That's okay.
Yeah.
How can the moisture in the algae be removed in terms of wet and dry biomass?
You're telling about wet microalgal biomass and a dry microalgae biomass or something like that. So you want to make the wet microalgae dry and you want to use it for something else? channels or something?
Yes.
Okay, so example, if you're growing on a media, like for example, Centrosmus obliquus grows well in BG11 media, blue-green media. So you're preparing that media, you're going to inoculate your algae into that media, and you're going to grow it, and you're going to scale it up. Like you start with a small, 50 ml flask, and then you scale it up. And once you've grown that algae, it's a wet biomass, right? So what we do is, if you want to make it dry, you want to get it in the form of powder, you just centrifuge it. At the end, you just centrifuge it, and you just remove the supernatant, and you just have that biomass.
Okay.
You freeze dry it, and you get it. Yeah, so that's the normal procedure.
Would adding fertilizer to the water help the algae grow?
Fertilizers, in the sense, like what type of fertilizers? Because algae generally, as plants, algae is also a plant. Like they say, it's a plant, but it requires nitrogen and phosphorus. Like those are the two most essential nutrients for growing algae. So it depends on what fertilizer you're adding, and in what amounts you're adding. So it could enhance, but I'm not sure of that aspect.
Would these additions impact the environment negatively?
Some nitrogen and phosphorus,
Right.
so it shouldn't, but I'm not sure, because it solely depends on the amount you're adding.
Right.
And also whether you're adding it in like you'll be adding it, and you'll be mixing it and dissolving it in something, right?
Yes.
So yeah, then it shouldn't be.
Okay.
Is algal fuel commercially viable, and can it compete with other types of energy resources?
As far as I know, we do have biodiesel in the US.
Okay.
So we do have biodiesel in the US, but yeah, algae sourced biodiesel, I guess. So right now, it's a bit costly than the conventional biodiesel, but if you're using it for some other purpose, like other than biodiesel, so it's more like a biorefinery concept, where you just don't focus on biodiesel. You try to enhance the biomass productivity, use it for biodiesel production, use it for a different source of purposes. For example, your algae which you're growing on, it will be rich in carbohydrate, protein, and lipid. Like it could be rich in either protein or carbohydrate or lipid, or it can have all three in different compositions. So if you're looking at carbohydrate, you can go for bioethanol production. If you're looking at lipid, you can go for biodiesel production. If you're looking at protein, you can use it for nutraceuticals like animal feed, because it depends what you want to get. So in that way, you can make it economically viable.
Okay.
So you can use it for a number of purposes. Like just biodiesel, I'm not sure whether it will be economic viable, but you can use it for a number of purposes, like producing value-added products, and then using it as a biodiesel. Because for biodiesel, you'll be just extracting the lipids, so you have the carbohydrates and proteins which is of no use. So you put it to some use, and it should be economically viable.
Would it be possible to genetically modify algae in order to create the most efficient species in terms of biofuel?
Species. Would that be
Genetically
possible?
modifying? Yeah, it is
For
possible.
algae?
Yeah, it is possible. Genetically modification studies are happening. At McGill, I think there's another lab which is doing genetically modification of some algae to produce biofuel, I guess. Yeah, so it's biofuel. So yeah, you can genetically modify algae to produce the desired amount of biofuel.
Yeah. And so now, basically, we were just wondering about what you were studying and doing as a chemist.
I am working on a microalgal consortia, so I'm looking at particular two different consortia, two different species of algae, strains of algae. Species is Chlorella species has a number of strains. So I'm studying strain specific. I'm taking two different strains. That would be a best way to say it. So I'm taking two different strains of Chlorella and Centrosmus. So I'm working on to improve its biomass productivity, and then I'm trying to use that biomass to produce value-added products, and also use it as a biofertilizer. Also use the treated, I'm trying to grow it on wastewater. So after extracting the microalgae, I use the biomass for value-added products, and I use the wastewater as a biofertilizer. So I'm looking at various concepts in the project.
Is there anything else that you could tell us concerning the genetically modified experiment that's going on right now at McGill?
No, I'm not. I'm really not sure of genetics, because I'm not I'm.
It's a different.
Yeah, it's a different area. It's a totally different area, because each strain has its own set of genes and set of genetic codes, and I'm not sure what they modify. Yeah, it's totally completely different from what I studied.
Even though genetically modified algae is not in his field of interest and a study, we think that it's a very interesting concept, because we know that we could increase the quality and increase the quantity of the biofuel produced if we can control the factors that improve this fuel and this algae.
So I read a study of someone that was using household equipment to break down the algae and try and create biofuel. What do you feel about that?
Household equipment.
So they were using blenders to break it down and then allowing the oils and lipids to separate, and then adding in NaCl, so something very simple and accessible, and trying to see what the results are from that.
Okay. I'm not sure of that study, but I'm not sure how it's going to work as well, because algae cells are you should either ultrasonicate it, because there are a lot of methods for extracting oil from algae. Apart from transesterification, you have first you have to break down the cells using ultrasonication. Then you do the transesterification, because this is done in order to ensure that you get the maximum lipid out of the algae. So I'm not sure how that works, but
okay.
yeah.
So you really need to be in the lab and use the equipment.
Use the equipment, yeah.
Okay, that makes sense.
How would you perform the ultrasonification in your lab?
You have an ultrasonicator here in our lab, so you just have to take the algae, the algae biomass, the wet biomass. You just have to put it in the ultrasonicator. I think it's downstairs.
Okay, so the machine does it.
The machine does it, so you don't have to do anything.
Okay.
So you just put it in the ultrasonicator. You just wait for 10 minutes, something, and then just do it. And if you want to confirm that, you can use a SEM, scanning electron microscope.
Mm-hmm.
That gives you an idea whether the cells have broken down or not. So you take a SEM image before you do the breakdown, and then you can do a SEM image. You can also do that as well. So, but I'm sure this SEM is a tabletop scanning electron microscope, and that is only for dry matter. So I'm not sure whether that can be done, but yeah. That's
okay.
In the sense of also extracting the energy source from the algae itself, we've encountered about two different methods, different from yours, which were gasification and pyrolysis.
So those are entirely different. Pyrolysis is a high heat, you give in a lot of heat and you just try to do that, right? So it's totally different from what we do here.
Okay.
And you were saying that the most effective is the method that you were describing.
Yeah, transesterification. Yeah.
And why is that again?
Why is that in the sense
why is it the most effective method to?
That is the most commonly used method.
Oh, commonly
used
okay.
method. So that's why.
So probably the most accessible and easiest.
Yeah,
to
the
perform.
easiest method for you to follow. Yeah.
Okay.
Went through most of our question. Would you have any leaving thoughts to add on?
Working on, so what are you trying to do in your project?
So just compare the different types of algae and the way that they produce the biofuel.
Mm-hmm.
Okay, so different types in the sense are you trying
Species.
to species?
Yeah.
Okay,
Yes.
different species. So are you looking at different strains? Because again, chlorella species is a broad term.
Now
You have
after-
different strains in that, like chlorella vulgaris, chlorella pyrenodosa. So those are different strains in that.
Okay.
So are you working strain specific, or you're working species specific? Because species specific is chlorella, ceratosmus, nanochloropsis.
Mhm.
All these are species, different species. But strains are nanochloropsis has nanochloropsis oculator. So many species are there under nanochloropsis as well. So it differs, what you're trying to do in that.
We understand we would be better off with species, since they will produce a more varied result, which would be good in comparison for us to deliver a report and results.
In between species, it's but
The difference is bigger.
yeah, the difference is bigger. But what I would suggest is even strains are different.
Oh, really?
They are really
Yeah.
different. Yeah,
Because
even
of the
strains.
lipid component,
Yeah,
yeah.
yeah. So
Yeah.
so strains are different. There's one table I could remember. Each strain, they have the amount of carbohydrate, protein, and lipid content. So you can go for the strains which have the highest lipid content. So in that way, you're successful.
Mm-hmm.
Because there is a table for that. So they have most commonly used algae strains. So they have that, and they have the percentage composition in dry weight. So
Okay.
dry mass. So
Yeah.
it will be useful for you if you.
Yeah, of course.
Yeah.
And how long does it normally take to extract the biofuel?
I've not done it personally, but it should take, it depends. but it's a lab procedure, so it should
Right.
take you like 1 or 2 days, except only thing is like you'll have to grow it. That process takes longer.
Yeah.
So extracting it just takes 2 days, 3 days at the max.
Okay.
Right.
But the process of growing
So
it,
it's
getting it
obtaining
done,
it and then doing
obtaining it
experiment
and
in the
doing
lab.
it takes a lot of time, because the normal lag phase, the log phase of algae is 6 days, 6 to 10 days.
Okay.
So that's the normal period, like where the algae reaches its maximum and then it tapers off gradually. So so you have to grow for 6 to 10 days, and then you'll have to inoculate it. You'll have to have enough biomass for biofuel, like extracting the lipids and then transestifying it to biodiesel. So you'll have to extract the lipids and then transestify it to biodiesel.
Okay.
So that's the normal procedure which you have to do.
So having the algae in hands, it's a shorter procedure, right? So it's
Yeah.
Okay.
So once you get your algae, once you get your strains, and doing the process is little, like, it's not complicated that much, except for the part where you have to you have to get that compounds, right? For example, saturated and unsaturated fatty acids. If you want to get that, you have to have a GC or something to analyze the components of it. So,
but I'm not sure our study will go this far though.
Right. I think we're really sticking with the basics just to show that algae is a viable option.
okay,
for
so you're
biofuel.
just trying to prove like different strains. So
viability.
like, so you can instead of going for species, you can just go for freshwater and marine.
Okay.
Freshwater is chlorella, cinerismus, and all those stuffs come under freshwater, and marine is Nanochloropsis. Then you have Junaliella, the Salina. So those all comes under marine microalgae. So you can compare 2 different habitats. Those are 2 different habitats as such. So you can compare that, and then you can narrow down to like species. So you have 3 different species. So you can also make a comparison with species and also based on their habitat where they're growing. So that will be like you have a 2-way like a discussion on that.
Would be great.
And were those still growing in North America, or those are something that you have to?
I think algae grows in North America, but I'm not sure what strain
which
grows in algae,
strain?
so I'm really not sure of that. So because I know that we get these algae strains from a culture collection, so they have all the algae like which is available. They have it. They just send it to you in a 10 ml tube, and then you have to transfer it. They send you the conditions as well, but those media are difficult to prepare. For example, marine, the default media for growing it is F/2 medium. So when you get your marine microalgae, you just transfer it into F/2 media, and that's it. You just keep on growing it until you get the required biomass for your biodiesel production or biolipid production, and then transfer it to biodiesel. And for marine, like for freshwater, it's BG11. The commonly used is blue-green media, and the media depends on strains as well. So, for example, chlorella and cinerismus, blue-green media is good. But if you're going like into specific strains, then you have to modify it. Like for example, the strain which I'm working on, for me MN8 is good. I tried BG11, it didn't work. So so if you're looking at chlorella and cinerismus like as a species, BG11 is good, and Nanochloropsis or Junaliella. These are the most commonly spoken algae strains which are like which are of prime importance. So so Junaliella, like they have carotenoids, high amount of carotenoids. Like if you're looking at Junaliella, so so it depends.
With the help of our expert, we've concluded that we will analyze various species of algae in different habitats. So we're gonna look at a species in freshwater habitat and algae in saltwater and marine habitat. And try to find a conclusion with the data we've encountered during our experiment.
And then I just have one more general question.
Yeah.
So to obtain the species or more specific strains of algae, I'm sure at McGill there's it's given to the program in order to help it.
You mean we have strains of algae?
No, but is it something that you have to buy? Is it something that's just
it's something you
supplied?
have to buy. I bought it for like 50 pounds, I guess. That was two strains.
Yes, kind of pricey, right?
50 pounds is not pricey,
No?
I guess. 50 pounds for 2 strains is it's pricey. It's like 25 pounds is like 1,000, 2,000. I'm not sure. Like it's like in dollars, I'm not sure how much it is, but
Okay.
So 50 pounds is for 2 strains. You will have to have 50 pounds.
Okay.
So that's in the UK. So if you get some from local sources, like you have some strain collection in I'm not sure, like University of Montreal or UCAM or doesn't know. Oh my God, I forgot the name of another university. What are the
Concordia?
universities? Like no, not Concordia. Like University of Montreal, another French university, like the
UQAM.
UQAM,
UQAM? UQAM?
UQAM,
Yeah.
and that's it.
That's it in Montreal.
Or
Okay.
there's U Laval in Quebec City.
In Quebec.
Yeah, I think so. So these
Yeah.
somewhere in these universities there is a strain collection. Like
Yeah, maybe in
that's
Laval, because I saw that.
yeah,
They
so
were.
they have a strain collection here as well. So if you get it from here, it's even cheaper for you.
Okay, that's good to know.
Yeah, so it's even cheaper for you. So it depends.
Okay,
Okay.
perfect.
Well, thank you very
I
much.
think we
no
asked all our
problem.
questions.